Okay. Welcome everybody to this H3D Foundation webinar. I'm Nicola Elliott-Wong, the Program Coordinator at H3D Foundation, and it's my great pleasure to host these sessions. Today we're really blessed to have a member of our GC-ADA family join us today and a great friend of H3D Foundation, Adrienne Edkins. And I'm going to kick us off by reading her bio. As always, there'll be I beg your pardon, notifications. As always, there'll be a Q&A session at the end. Okay. Adrienne Edkins is Professor of Biochemistry and holds a Tier 1 DSTI NRF South African Research Chair. Adrienne completed her PhD with Bill Cushley at the University of Glasgow on a Wellcome Trust Four-Year PhD fellowship. Adrienne is NRF-rated and has been recognized for her research through the Rhodes University Vice-Chancellor's Distinguished Research Medal in 2015 and the DSI South African Women in Science Award for Distinguished Young Scientist in the Natural Engineering Sciences in 2018. Adrienne is a Fellow of the Royal Society of South Africa and the African Academy of Sciences, Fellow and President-elect of the Cell Stress Society International, and a member of the Academy of Science of South Africa. Adrienne's research focuses on understanding mechanisms of protein homeostasis in health and disease and applying this fundamental knowledge to infectious and non-communicable drug discovery. Adrienne, we are so thrilled to have you. It's been a while in the making. We've been bugging you to have you on, so thank you so much for joining us. I'm going to hand straight over to you, and everyone populate the chat with your questions, and we'll address them towards the end of this session. Thank you so much, Adrienne.
Wonderful. Thank you, Nicola. I really appreciate that introduction. I'm just going to share my screen. Okay, just minimize that. All right, can everybody see the screen?
It looks great.
Wonderful, and you can hear me.
Yes, perfect.
Okay, super. Thank you so much. It's an absolute privilege to be here today. I'm really fortunate to be able to share some of our research with you. So just by way of introduction, I will say a little bit about the group and the people whose work I'm going to be discussing today. So we're a research unit based at Rhodes University, and we incorporate the South African Research Chair. And we're also members of two consortia, one which is through ARUA and the Guild's Centers for Research Excellence, and the other, of course, which is the Grand Challenges Africa Drug Discovery Accelerator, which we are very fortunate to be able to be part of and allows us to interface and get lots of support from both the H3D Foundation and H3D itself. So if you're wondering where we are this little purple star. If H3D is down in the Cape, we are in the Eastern Cape, just about an hour and a half in from the sea. And essentially, our research is focused largely around understanding protein biochemistry. So proteins are the major structural and functional units of cells. And so they're implicated in cell health. And this obviously has consequences then for disease. So we take a protein-focused view to try and use cell biological and chemical biological processes to understand how proteins regulate cells. And so today, I'm really privileged to speak to you about some work that we've done in a virus called Kaposi sarcoma-associated herpes virus. And the work has been done by some really talented researchers in my group. So Dr. Michaela Valtain started off the work as part of her PhD, but the vast majority of work was done by Elisa and Michael, who are currently postdocs in the group. And then I need to just acknowledge Clint Veal, who's a long-term collaborator of mine, and Adrienne Whitehouse from Leeds University, who was really integral in helping us get the project up and running. So what's KSHV? So KSHV is a human herpes virus. So it's in the same family as viruses like EBV, Epstein-Barr virus. It's a double-stranded virus, and it's recognized as a type 1 carcinogen. And that means it's necessary, but not sufficient for the development of cancer. The virus itself is associated with 3 malignancies. Okay, 2 of these are lymphoproliferative disorders, so they're B-cell diseases, and one of them is a solid soft tissue tumor, Kaposi sarcoma. And Kaposi sarcoma itself can be one of 4 different types, but all of these different types generally arise in the context of some sort of either genetic or immune suppression background. And if we look at the distribution of KSHV infection, and we then align that with the malignancies, we can see that KSHV is actually not equally distributed geographically. So it appears to be higher levels of infection in certain countries, and this then correlates with development of higher levels of these tumors. And in particular, although there is a well-described classical Mediterranean type of Kaposi sarcoma, the extremely high prevalence of this virus in African countries has resulted in the major burden of these cancers being felt by African populations. And this is only unfortunately anticipated to grow with time. So unfortunately, there are somewhat limited treatment strategies. So there are things like immune reconstitution. So when you support reconstitution of the immune system in the context of HIV-associated KS, you can see some reductions. But this type of therapy is not effective for people who have KS as a result of other forms of immune suppression. So really, patients are left with the option of chemotherapy, which is not really very effective, and there are no specific targeted antivirals against KSHV. So we saw this as a real opportunity for us to not only do some interesting work in the molecular virology space, but to particularly apply this now to drug discovery efforts. And at the time that we started working in this area, there were relatively few people on the continent working on KS itself, on KSHV, molecular virology, and particularly on drug discovery. So the research program that we devised was really to say, well, can we use viral proteostasis as a target? Okay, and could we come at it from 2 different angles that would give us the best opportunity of trying to get something that was effective? So could we use targeted drug discovery approaches? So establish the techniques for molecular virology to understand how the virus interacts with its host, and in doing this, identify critical nodes for intervention. And then focus on these host and virus-based therapies using 2 strategies that we felt was going to be most successful. One, to use drug repurposing. So if we could the particular processes that were being harnessed by the virus in the host, could we repurpose existing drugs? And then could we look at undruggable targets? And this is really around trying to expand the druggable space and to use technologies that are now available to allow us to go beyond traditional enzyme-based inhibitors. And then obviously recognizing that phenotypic drug discovery also can contribute substantially towards identifying hit molecules. We decided that we would also integrate this, and we would try to develop a robust pipeline for hit discovery for anti-KSHV antivirals, use this to screen a series of chemical libraries, and then take forward any hits from that in a preclinical analysis. And so really the aim is to both establish and develop the tools to work with the virus and then apply them in these different ways to drug discovery. And essentially, I will tell you 2 stories. Obviously, these projects are still ongoing, but I will tell you 2 stories, one from the targeted angle and one from our phenotypic study. But before we go there, I need to give you a little bit of background on KSHV and how it exists. So the virus essentially forms a latent infection in B cells. So often this can happen in childhood. Children will acquire the virus perhaps through salivary contact, and they will have this latent infection in their B cells. And what we have here is a viral episome. So KSHV doesn't integrate, but it tethers itself to the host genome. And in that way, it establishes this persistent latent infection where it is transcriptionally silent, so very few genes are expressed, and the genome is essentially copied along with the host genome. But in certain environments with particular stimuli, we will get lytic replication. And lytic replication is when the virus now produces all of its around about 80 genes, okay, so approximately 80 proteins and a couple of microRNAs. And essentially, this kicks off DNA replication. It kicks off translation to produce the proteins that are needed to make new viruses, and you'll get release of these infectious virions into the circulation. These virions can then go on, infect endothelial cells, and result in transformation into the cancer that we know as KS. However, this cancer is obviously rapidly dividing. That's a hallmark of cancer. And because the viral genome is not integrated into the host genome, there is a chance that you will somehow lose the genome during that rapid proliferation. And so what you find is that there are periodic episodes of lytic replication and reinfection that maintains this viral genome within the population of the tumor. And so importantly, unlike some other herpes viruses, we understand that both the latent and the lytic cycles of KSHV are required for tumorigenesis. And therefore, our therapeutic approach has to really take into account both of these. And in particular, given our interest in protein homeostasis, we have chosen to focus primarily on lytic replication. And if we look at the process of lytic replication by KSHV, we can see that it's an extremely regulated and ordered process. So in latency, which is this persistent infection, we have a few genes that are expressed, and one of the classic ones is LANA. This is responsible for tethering the viral genome to the host genome. So it's absolutely required for latent or persistent infection. When you now reactivate lytic replication, the first gene produced is ORF50. So this we would call essentially the lytic switch that occurs as you progress from latency into lytic replication. And then what we have is a carefully controlled temporal production of viral genes, okay, and proteins, starting with the early and the delayed early genes, which encode often the replicases and the proteins, enzymes, viral enzymes required for the cell to make RNA, to make proteins, etc. And then at the later phases, you start to get the production of late proteins, which are things like your capsid proteins required for assembly of your virus. So this essentially transition between latency and lytic creates a high proteostatic burden in the cell. So essentially, viruses must be able to cope with this within the host cell. And so we were particularly interested in coming at this because of the proteostasis, the interest we have in proteostasis. Just to reiterate, proteostasis is a term that we use to refer to the collective processes that govern protein function in cells. So proteins will be translated as a polypeptide chain. They must fold into three-dimensional structure. They will then fulfill their function, and when they're no longer needed, they'll be degraded. We also have the capacity to maintain proteostasis. So this happens under normal conditions, but we can maintain proteostasis under essentially stress conditions. The problem arises in disease, where often the folding need or the proteostatic needs of the cell exceed the capacity. And as a consequence, we now get into disease. And essentially, all of these non-communicable and infectious diseases are impacted by defective proteostasis. And I will give you the example of how this is done in viruses. So viruses essentially are intracellular obligate pathogens, so they must infect a cell in order to replicate themselves. And normally they will carry on their genomes the absolute minimum genetic information required to establish that infection. And this means that they co-opt and they use host systems to fulfill many of their biological functions. So in our case, we know that molecular chaperones, so these are proteins that are responsible for maintaining proteostasis, are hijacked essentially by viral proteins. So you have a host proteostasis machinery that KSHV, for example, will need to use in order to be able to fold its proteins, in order to be able to establish that lytic replication process, in order to make new viruses, and then to go on and be oncogenic. And if you look at this diagram that I have, this is just a representation of some examples of the types of molecular chaperones that we have in the cell. Many of them are heat shock proteins. And the point of this is to demonstrate that they function essentially as networks. So none of these systems work on their own. And in fact, each of these systems is a complex network of protein-protein interactions. And we also recognize within these networks another type of protein, which is called a co-chaperone. And essentially, this co-chaperone would be a protein that lacks its own chaperone activity, but can regulate the activity of another chaperone. And this is essentially where we will focus the talk now. So we were interested understanding this lytic profile of KSHV and the potential proteostatic burden to have a look and see how a target-based approach would be towards inhibiting the virus. And essentially, we knew from other studies that this molecular chaperone HSP90 and HSP70 were both required for KSHV biology. These proteins are both proteins that have been targeted as potential targets in the context of other diseases. And so we had available some existing inhibitors. Both of these inhibitors work on the basis of inhibiting ATPase activity. And what we could see is that, yes, in the latent phase, both HSP90 and HSP70 inhibition was effective at clearing KSHV-infected cells, but that we observed substantial resistance during the lytic phase. And so we thought that perhaps we must investigate an alternative approach to targeting these two systems. And could we do this using a focus on the co-chaperone? And that's this protein here called HOP. So HOP is a modular protein, so it has repeating units of these different domains, and it has selectivity for which chaperones it binds. So it will bind HSP70 at an N-terminal site, and it'll bind HSP90 within the middle. And of relevance to this talk is essentially this TPR2A domain. So we know that HSP90 is involved in KSHV biology. We know HSP70 is involved. And we know that essentially this protein HOP is responsible for getting HSP90 to talk to HSP70. And so we ask ourselves, well, can we then evaluate whether HOP plays a role in KSHV biology and use HOP as a target to simultaneously take out these chaperone systems? However, at the time we did it, there were no well-defined inhibitors. And so we started out essentially using a genetic approach. Okay, so can we use shRNA, so genetic depletion or overexpression of HOP, to evaluate whether it actually is core for these viral processes? So here we used some cell line models. So we have a B cell cancer called BJAB, which is not infected. So it serves as an uninfected model. We have a BCBL, which is a KSHV positive cell line, and it essentially serves as the latent model. And then we have a modified version of the BCBLs. And this cell line you can use to drive high levels of lytic replication by doxycycline-regulated expression of OR50, which is that first lytic switch. So we use these models to essentially validate whether HOP is a potential target. So we can ask ourselves, well, if we take our uninfected and our infected cells, do we see a change in HOP? And while we see an upregulation in both the HSP90 and HSP70 levels, HOP levels didn't change. However, when we now took our infected model and we pushed the cells from latency into lytic replication, we could see a significant increase in both HOP protein levels and at the HOP mRNA level. So this suggested to us that HOP was being upregulated in response to the requirements of lytic replication. In addition to that, we can go in and look at whether HOP is involved in the lytic cycle. And to do this, we did immunofluorescence here. So we are showing you these BCBL cells where they are either in the latent form. Okay, and here we have HOP shown in green, LANA shown in red, the nucleus in blue, and the coincidence of the HOP and the LANA staining, which reflects basically co-localization of the proteins being shown in this merged image. And what we could see from this is that in the latency, the HOP puncture and the LANA puncture were almost adjacent to each other. And as we induced lytic replication and we monitored the cells over time, we could see an increasing co-localization between LANA and HOP. So LANA is this core protein that is essentially a marker of the KSHV viral genome. And at the late time points, we could see a phenotype here that essentially reflects what are known as replication factories, with HOP being intimately co-localized with LANA and the viral episome during that replication process. So this said to us that HOP is potentially involved in this lytic process. So now, can we validate that using a genetic approach? So we started first by doing HOP depletion. So here we made stable cell lines with either a non-targeting control or an shRNA against HOP. We're able to get very successful knockdown levels of HOP and then use these cells to study what happens to the lytic cycle. And what we can see is in certain cases, so not all genes are affected, there appears to be a selectivity, but depletion of HOP significantly reduces viral mRNA transcript abundance. This then translates into a reduced viral load. So this is the amount of viral DNA that you have as a function of the host genome. And it results in a reduced level of infectious viruses. So this was suggesting to us that depleting the levels of HOP is going to be important for lytic KSHV replication. We then did the opposite experiment. So now we can go and put in GFP HOP. So we can overexpress it. We can artificially elevate the levels. And then we can ask, what do we see in the context of lytic replication? And in this case now, we see that for selected genes, again, we see an increase now in the production of viral mRNA transcripts, again, translating into a greater viral load and an increase in the infectious viruses. So this to us said, well, we're now pretty happy. We can see that HOP is potentially a new host factor for KSHV lytic replication. But is it a druggable target? So this really was quite a challenging question because HOP actually lacks any inherent ATPase or enzyme activity that we could easily target with a small molecule inhibitor. And so this is where we turned to our collaboration with Clint Veal, because HOP essentially functions mainly via protein-protein interactions. And so essentially, if we were going to develop an inhibitor, we were going to need to develop something that could block a protein-protein interaction. And so we moved then to a crystal structure that was available that was essentially defining the interaction site between that TPR2A domain and HSP90. So this is the domain peptide interaction. And we wondered whether we could use this peptide as a way to start to develop inhibitors of this interaction. And so this is where we worked with Clint to use his native mass spectrometry approaches, where essentially you can take a protein and its ligand, and you can combine them and infuse them directly into the mass spec, and you can identify both the APO protein and the protein with the ligand bound. And so essentially, Clint was able to develop a native mass spec method here based on our target site and the native peptide ligand. In parallel, we had used a focused acidic fragment library, because obviously we have two glutamic acids here that are important for the binding to TPR2As. So we had, in parallel with this peptide analysis, we had screened an acidic fragment library for those that bound to hop. And we found that many of the fragments that bound to hop were actually enriched in tetrazole moieties. What I know now, and I didn't necessarily at the time, is that the tetrazole is actually a bioisoster of carboxylic acid. And so what we could do now is replace these carboxylic side chains within the peptide and see whether that would now impact the binding to TPR2A. And what we could find was that, yes, substitutions of these carboxylic acids with tetrazoles resulted in robust binding to TPR2A. And in fact, when we then took this into a protein-protein interaction model in vitro, we could see that compared to the native ligand, which is here, both these substitutions of a tetrazole-containing glutamic acid in both positions were very effective at disrupting the interaction between HOP and HSP90. And based on the modeling, we think that that's probably due to the acquisition of a new interaction site here with lysine residues within the TPR2A protein. And then we also did some additional studies where we confirmed that these peptides were selective, that when we tethered them onto a resin, we could actually pull out HOP from lysates. And in fact, we could actually pull out specifically only the TPR2A domain, and it didn't bind to any of the TPR1 or the TPR2B domains. So then the question was, well, do these peptides have any anti-KSHV activity? And so now we return to our model. And so we have 3 of these tetrazole-containing peptides. So just to bring you up to speed, we have a double tetrazole, and we have one with a single one in this position that are both equally effective. So you will see some of the data is referred to the double one, and some of it will be with the single one at that position. But essentially, we could see very potent inhibition of the interaction. But when we now went into our activity assay, okay, so here we monitor lytic replication by the absence of this protein in latency and its presence in lytic replication. When we compared the vehicle treatment with the peptide treatment, we saw absolutely no change. So of course, this was suboptimal, to say the least, and we wondered whether perhaps this peptide wasn't getting into the cell, and that's why we weren't seeing the effective inhibition. So now we were able to tether it to a CPP. What we found was that CPP at low concentrations still disrupted the protein-protein interaction, but it stabilized it at high concentrations. So then we had to show that that was due to the fact that that CPP is causing that stabilization. And in fact, when it gets into the cell, so I neglected to say that the CPP was linked via disulfide bond to the tetrazole. So it would effectively carry the tetrazole containing peptide into the cell. And then when the disulfide bond is broken, it would release the peptide. And we could see then by adding in DTT to break that disulfide bond, we could actually recover the inhibition. And so when we did that, now we could start to see a dose-dependent reduction in the levels of these lytic proteins. And again, if we then took that into some of our other assays, we could see that this peptide also now reduces the gene expression of a host of lytic genes, being most effective against the late lytic genes. And similar to what we'd seen with our knockdown, we were able to see that these peptides could significantly reduce viral load, significantly reduce the production of infectious viruses, but most importantly, show limited toxicity towards the cells. So really, we are quite happy now that this represents chemical validation of HOP as a drug target against KSHV lytic replication, and demonstrating that we can, in fact, somehow modulate that proteostasis target in a way that the virus becomes susceptible, but the host cells are able to retain sufficient proteostasis capacity in order to survive. So this part of the project is obviously carrying on. We're trying to do some, we've done some global proteomics analysis. So we have now a viral specific and a host specific proteome that's affected by HOP knockdown. And we have a host specific HOP interactome that we've done through proteomics. And essentially, we're now going to use these two data sets to move forward. One with trying to see whether we can change that peptide into something that's more druggable, but also to use the proteins that are influenced by the loss of HOP as candidates for drug repurposing. So if we know that HOP knockdown influences inhibition of the lytic replication, can we define which of those host proteins is responsible for that inhibition? And ideally, can we then find an inhibitor against that protein that would then allow us to repurpose existing drugs? And so that work is ongoing, and hopefully we'll have some stories to report about that in the future. So now I just want to, before I finish up, just to tell you a quick story about the phenotypic screening component of this project. And essentially, here we were interested in setting up this pipeline. So we wanted essentially a library that we could start with, and we were very grateful to MMV for providing us with a pandemic response box. And essentially, we used this as a pilot to demonstrate our phenotypic screening cascade. So in a single image that really reflects an entire chapter within Michael's PhD, he was able to develop this lytic replication cascade. So where we would induce lytic replication, we would treat with a single point of the inhibitor. We would screen out any compounds that had cytotoxicity greater than 70%. So this would be done in parallel. And we would then be able to use an ELISA to measure the levels of this lytic protein. So ORF57, this is an early lytic protein. We chose this because there's a very good antibody for it, and it occurs as an early marker. So it gives a good sensitive readout for KSHV lytic replication. We could then normalize that to total protein and use essentially this process here to triage our compounds. So Michael put the 400 compounds from the MMV pandemic response box through this. We then came up with 7 compounds that passed our initial screen. He was then able to do some confirmatory screens, including treating at different lower concentrations, looking at readouts like viral mRNA, viral DNA load, and infectious viruses. And that led us to one compound that we now know has excellent anti-KSHV activity. And so this compound we are now progressing through as a hit from this phenotypic screen. So here we can see the results of the confirmatory screen. So we now do this in a Western blot format so that we have an orthogonal validation for our ELISA assay. We can see again reduction in the levels of ORF57 with this molecule, but not these other molecules that were initially identified as hits from the screen. So we've kind of parked those for now. We could also see in another cell line model of KSHV lytic replication that this compound was effective. So we know that it's not just related to a doxycycline phenotype. And so we really now progressed with MMV, I'll just call it 152 from now. We're able to do now a dose-dependent analysis here to see that if, in essence, the loss of ORF57 is dose-dependent with the increase in 152, we were able to source 4 different analogs commercially available. And interestingly enough, we identified, obviously here's the hit that's showing inhibition here, and we could see that compounds 1, 2, and 3 were actually inactive, and compound 4 we refer to as our partially active control. So this was quite nice because it meant that we also now had chemical material that we could use as controls. And so where you see in my subsequent slides 1, 2, or 3, or 4, these refer to either an inactive compound or a partially active one, as it speaks to inhibiting KSHV lytic replication. We were also able to confirm that this phenotype was recapitulated both in a model that is closer to the endothelial model for KSHV infection, and in one where you have both KSHV and EBV co-infection. So again, we think we're not getting cell type specific effects, but this is related specifically to the molecule in question. So again, Michael went on to confirm that we could see activity in our various antiviral assays. So again, this is a gene expression assay. So we're looking at lytic genes. So we can see that there's no gene when the cells are in latency. You induce lytic replication with doxycycline, you get a substantial increase, and we can get a dose-dependent inhibition of the production of this transcript with our hit compound 152, but not with one of the inactive, oh, sorry, I beg your pardon, but not with one of the inactive analogs. So here's analog 1. Again, you see that same increase with lytic replication, but you don't see any suppression. And the same was true again for ORF65, where we see a potent inhibition, but not with our hit compound. And again, Michael surveyed a number of other genes, and so we could see that for many of these genes, the genes that were affected by 152, the inactive or partially active analogs didn't give a significant response. Okay, and so then again, we looked at the viral load, and again, we can see that, again, now measuring the amount or proportion of viral DNA relative to the host genome, we could see an increase with lytic replication, and then a dose-dependent increase with the hit molecule that was not then recapitulated by either the inactive or the partially active compound. And then last of all, Michael tested around virion production and infection. And again, we could see that in both of these assays, so one where you treat the KSHV infected cells, okay, and you see how they produce infectious virus that can infect the HEC cells, or one where you leave these cells untreated and you treat these cells. And essentially we can see in both of those cases, essentially ORF152 is effective at blocking the production of infectious viruses. It has a minor but non-significant effect when it's used treating the HEC cells. So we really are now at a point where from this initial screen, we've been able to develop the screening cascade, and we've really been able to validate it now with an identification of a hit compound. We're now working with Clint again. So we've done a number of structure-activity relationships that have allowed us to actually develop an analog that has increased potency. And we are working now on the preclinical validation of the target of this molecule and the mechanisms of actions, as well as trying to prioritize some in vivo models and ADME analysis. In addition, we're now consistently running the screen for anti-KSHV hit compounds using this phenotypic approach. And so if there is anybody who's interested in collaborating in this and would like us to screen their compounds, you're welcome to get in touch with us. So it just remains for me to say then a great thank you to both the collaborators, but also the people who did this work. So again, Elisa and Michael, who were really integral in setting up the KSHV technology in our lab. Adrienne Whitehouse at Leeds, who's an expert in KSHV biology, and then obviously Clint, who's really brought a huge wealth of expertise to the project from native mass spec all the way through to chemical synthesis. And then just to again acknowledge our funders, obviously MMV for the kind production of the pandemic response box, and then obviously to H3D and H3D Foundation and the Grand Challenges Africa Drug Accelerator that have really helped us to develop this project under that platform. And then obviously, you know, they say it takes a village to raise a child. Looking at this slide, I think it also takes a village to fund a research project. So I'm just very grateful to all the agencies that have given us the funding that's allowed us to really take this project through to a successful approach. And so with that, I will say thank you very much, and I'm happy to take any questions.
Adrienne, fantastic. Thank you so much.
Thanks.
Even I was able to follow along. I have no intelligent question, although I have plenty of intelligent questions, but I think we're going to have some questions from the group. And I'm going to ask so I don't know how to pronounce your name, Moketli, Morake, please go ahead and unmute yourself if you'd like to ask your question in person. Let's see if you're up to doing that.
Hi there. Thanks, Adrienne. It's quite interesting to see the data and the work that you're doing. But I was just wondering on the proteomics data, what was the concentration of the compound tested and whether there was any significant reduction in terms of the HOP expression that you spotted? And the second question was really more on the disulfide bond cleavage, right? Is it possible that, you know, after the compound has been cleaved and it's this free thiol, that it probably interacts with the glutathione inside the cells? And what are the levels of glutathione inside those cells?
Thank you. Those are excellent questions. So just to address the question around the proteomics. So we haven't done the proteomic analysis on the inhibitor-treated cells. So the proteomic analysis that I was referring to is the knockdown. So essentially, that was essentially establishing what's the hallmark of the knockdown, so that we can essentially try and identify the mechanism of HOP's function. But you're right, we could look in there and use some of those markers to validate, for example, in a Western blot, the response of our inhibitors. Yeah, and then the question on the disulfide bond is absolutely relevant. I must admit, I don't know what the glutathione levels are in there, but I will definitely be looking it up after this talk, and I think that's great insight that we can follow up on. Thank you.
Thank you very much. Yeah.
Thank you. Fortunate, would you like to go ahead? And guys, please feel free to raise your hands so we can get an order going.
Awesome. Thank you so much, Adrienne, for a brilliant presentation as always. I wanted to find out at the beginning there where you are seeing a reduction in the levels of HOP, but not HSP90 or HSP70. Do you think that there's a different mechanism of action at play here, where HOP is interacting with viral proteins in a manner that's very different to our classical proteostasis understanding?
Thanks, SAWiSA. Hi, and that's a really great question. So I think you are absolutely right. So we know that in the field of proteostasis, there's kind of this dogmatic idea that the role of HOP is really only to facilitate this interaction of HSP90 and HSP70. But we definitely believe that that HOP has its own independent functions. And I think that the reason that we're seeing the phenotype with a molecule that essentially is designed to disrupt the interaction of HOP and HSP90 is because this is the only or the first inhibitor of this type that actually binds to HOP. So there have been some inhibitors where they've tried them in the past, but they've designed them against HSP90. And so I think that is perhaps also telling us that we've got some sort of different effect. Again, if you look at the HOP knockdown studies in some of the uninfected mammalian cell line models where the work's been done independently of viral infection, you see very few changes to the proteome, even though you do see changes in biological functions. So we suspect that maybe HOP's role is more about specific types of functions, and those types of functions are the ones that are needed predominantly by the virus, and the compensatory pathways that the host cell can use are not actually available to that virus. So it's just a case, obviously, now of proving that. So that's a harder thing to do.
Fantastic. I look forward to hearing about the HOP co-chaperone.
Thanks, Forge.
Jackson, would you like to go next? Okay, I'll read out his question. On the PPI interaction, how does the TRPV proteins modulate the viral infection?
If I understand correctly, are you speaking towards how this actual domain of TPR2A is involved in coordinating the virus biology? I'll answer that question. I hope I've understood it correctly. So that's one domain that is predominantly involved in a particular type of interactions. So from our interactome data, okay, we did a scenario where we immunoprecipitated HOP at different phases of the lytic cycle, and we were able to see the acquisition of new interactomes. So essentially in latency, we could see HOP interacting with host proteins and viral proteins in complexes. And then as we monitor those complexes over the process of lytic replication, we could see host proteins dissociating, viral proteins associating. So what we think is that HOP is essentially a scaffold. It's sitting there as a scaffold with these different binding domains, okay? And you have different specificities at those domains. And what that means is that it can coordinate formation of complexes. So a certain protein will bind at this site and another one will bind at that site, and that will then facilitate the interaction between those proteins. So that's how we think it's doing it. There's probably going to be a role for the chaperones in there, but I think that the sites that are involved in chaperone binding, in other words, binding to HSP90, are going to compete with the viruses. And so another outstanding question is, how do we understand how the virus competes with the sort of natural interactor at that site, and whether we could subsequently use that as a tool to develop even better inhibitors.
Okay, you were honored with that answer, according to Jackson. There's a follow-up question. Do you presume a possibility of dynamic PTMs at play?
Absolutely, yes, absolutely. So we know that post-translational modifications are really, really integral in terms of regulating the biology of these proteins, and HSP90 and HOP are no exceptions. There are a number of papers that have looked at changes in HOP function and showed how affinity is linked to particular post-translational modifications. And so we absolutely predict that could be it. That could be a way of regulating the specificity for one protein over another. We just haven't done any experimental work for me to be able to give you a specific example in that case.
Okay, fantastic. Okay, enough about the science now. Let's get to the real question. The real question, guys, please raise your hands. If you have more questions, no worries. But I want to ask, how do we, at GC we're always looking at how do we combat the brain drain? How do we keep scientists like you on the continent? What are we doing right to keep you here? And what could you, maybe the follow-up question, what advice would you give to younger scientists who are wanting to carve, a pathway in science in Africa? what do they need to keep in mind? So it's quite a broad, open-ended question. Do with it what you will. But yeah, that is my question.
So I mean, it's a great question. I mean, I don't know if I'm qualified to answer it, but I will do my best. I see science in Africa as a massive opportunity. I think that we have not even begun to really harness the potential that we have. But I agree with you that if we don't create an environment in which people can do high-end, intellectually challenging work, then inevitably they're gonna go somewhere else. So for me, part of this project was really about creating a platform for skills and technologies that mean that we can keep this, we can do this work here. So we can do the kind of drug discovery work that is needed by people in Africa, but we can lead it, and we can do it here. And obviously, I've been extremely fortunate. So linking in with GC-ADA has been instrumental in terms of giving us the support and the networking to help us really push this project forward. I think that we are sitting at a point where we are very close to developing critical mass on the continent, and that really is going to catapult the research into the next phase. And so I would just say to people, it's not necessarily better somewhere else. You're going to trade whatever issues you might have here for a different set of issues, and that this is a really exciting time. There are many groups that are doing really exciting work, and we want you to stay here and be part of it. I think also there's a real opportunity for biotech. I think that sort of space that is still developing in South Africa compared to the global north. I really think that there's an opportunity to learn from those experiences and to fast track and be innovative on the continent. So I hope that answers your question.
Yes, and what a lovely optimistic message for our almost end of the year. Thank you, Adrienne. And collaboration. I mean, collaboration is king,
Absolutely.
isn't it? Right?
Absolutely.
And I think you've emphasized that in your presentation as well.
Absolutely. Yeah, and exactly. Nobody gets anywhere on their own. And again, if you can find somebody with whom you can have a great working relationship, then that's an extremely motivating and supportive thing. Yeah.
Okay, so guys, I've populated the chat with our usual HCD Foundation links. Please do connect with us. And also the feedback form, if you have great topics in mind or speakers that you'd like to propose, or even to give our speaker today or any speakers so far feedback or kudos, you please do so. We love to hear your feedback and get your input. Okay, so and then just to say a huge thank you to Adrienne for this fantastic presentation. It came through so smoothly with such great execution. I'm really just blown away by it. And thank you for the amazing work that you are doing and all your contributions to our projects. We are so grateful to have you on board. Thank you.
Thanks, Nicola. Keep well, everybody.
Okay, everybody. We'll see you next time. Have a great week.
Bye.
Bye.
Bye, everyone.
Have a good time. Bye.